Chapter 7, Blood Screencast 4. Now, this is the last screencast for Chapter 7, and we'll be focusing on a few things. The main topic that we're going to start off with are blood types. Now, a very common medical procedure is a blood transfusion, and this is when the administration of blood directly into the bloodstream of a donor. Now, depending upon whether or not this transfusion will be successful is based on matching the blood type of the donor with that of the recipient. We use what's known as an A-B-O blood type. So this gives us the options of you can be type A, type B, type AB, or type O. Now, there's also one other antigen, the Rh antigen. So for instance, my blood type is A negative. What that means is I do not have the Rh antigen on my blood cells. If I were A positive, then that would mean that I actually have the Rh antigen on my blood cell. Now, what's important for you to completely understand the blood typing is the concept of antigen versus antibody. Now, when we talk about an antigen, that is a non-self protein, so it's foreign to the body. It may be on a cell from another individual, or it could be coming from an invading microorganism. Now, antibodies, these are defensive proteins that are produced by the body, and they typically target specific antigens. Now, when we looked at plasma, one of the plasma proteins were these gamma globulates. So they're found in the plasma. Many antibodies are directed against specific infectious microbes. Other antibodies may be directed against other antigens, such as those found on the cells of blood received in a blood transfusion, or antibodies may clump and actually inactivate antigen-bearing cells. So here we can see that process going on. So the antibody binds to the antigen, we can see that here. And notice the antibodies are ignoring the self-surface proteins. They bind to the antigen of the foreign cell. So that's important. Then notice how the antigen-antibody complexes clump together. Now this clumping, or agglutination, which is the actual name for that process, basically inactivates the foreign cells. Now here we can see type A, which means they have type A antigen, type B antigen, AB, so both A and B antigens are on the red blood cell, and type O is when neither antigen is on the red blood cell. Now, when we look at antibodies, if you are antigen A, you will have anti-B. If you are blood type B, you will have anti-A. If you are type AB, you will have no plasma antibodies. If you have type O, then you will have anti-A and anti-B. So something to keep in mind when we're talking about various blood typing. Now, ABO blood typing is based on the A and B antigens. So this is something to keep in mind here. Recipients must not receive cells that they have antibodies against. So someone with type A, because they have anti-B, can receive type A or type O blood. An individual with type B blood has anti-A antibodies, and they can only receive blood type B or type O. Now, those individuals that have AB, they have neither antibody, and they can receive A, type B, type AB, or type O. Now, the individual with type O blood, they have anti-A and anti-B, and they can only receive type O. Now, what's important here, remember, type O does not have any antigens. Because there are no surface antigens in type O blood, it's not going to create or stimulate an immune response. So we consider type O to be the universal donor because everyone can get type O blood. Now, the other factor is the Rh. Remember, I talked about that previously. That's another antigen that can be found on red blood cell surfaces. The majority, actually 85% of Americans, have the Rh antigen, so they are considered to be Rh positive. The remaining 15% are Rh negative. Now, this is important, okay? People that are Rh negative will only get anti-Rh if they have been exposed to Rh type blood. So that's important. So they do not naturally have anti-Rh. They have to be exposed first. And the most common example of this is Rh incompatibility during pregnancy. So let's take a look at this. So here, let's look at the situation. The father's Rh positive, the mother's Rh negative, and now the developing fetus is Rh positive. Now, the first pregnancy is not an issue because at this time, the mom does not have anti-Rh. Now, during pregnancy, or more commonly at childbirth, a small amount of fetal blood enters the mother's circulation. Over the next several weeks, the woman develops antibodies and an immune memory against the Rh antigen. When the woman becomes pregnant with her second Rh positive child, her immune system quickly produces antibodies that attack the fetus's red blood cells. So that's the issue. So these women are typically given an injection called Rogan, and what that does, it actually depresses or suppresses the antibodies so the pregnancy can go to term. Now here, this is how we actually determine an unknown. You look for clumping. And so I used the term agglutination previously. And so we can actually see some examples of the clumping, okay? And so the agglutinated type A contains antigen A. So notice when anti-A was added, it reacted, so we know that that is type A blood. Anti-B, nothing. Type B, okay? It has antigen B, so we added anti-B. We see the agglutination, so we know that we're talking about type B blood. If we have type AB, then they are all both going to clump, okay? And then if I have type O, agglutination or clumping will not occur in either of the unknown samples. Now, there are things known as blood substitutes. Now, there may be transmissible diseases in donor blood in which a substitute might be necessary. The right type of blood might not always be available. There may be limited storage of whole blood or an inability to meet the demand for donor blood. So that is why there is the need for blood substitutes. Two types of oxygen-carrying blood substitutes. Various modified hemoglobins in aqueous solution. The hemoglobin must be packaged, though, in some manner. Perfluorocarbons, or PFCs, in aqueous solution can actually carry more oxygen than whole blood. Now, we're going to be looking at a number of blood disorders. The first infection is mononucleosis, and this is due to the Epstein-Barr virus infection of lymphocytes. It's also known as mono. You may have heard of this as the kissing disease, and typically it presents with flu-like symptoms. Blood poisoning is actual bacterial infection of blood and known as septicemia. This can develop from wounds that become infected, burns, a UTI, or actual big dental procedures. Now, blood poisoning can be life-threatening, and this is something that does require antibiotic treatment because we are talking about a bacterial infection. Now, anemia, that is a generic term that talks or alludes to reduction in blood's oxygen carrying capacity. So there's a lot of different things that can actually cause anemia. Now, the most common is iron deficiency anemia. So these individuals are not getting enough iron, or they may not be absorbing the iron properly. Hemorrhagic anemia, that's caused through blood loss. Pernicious anemia, that's caused by a vitamin B12 deficiency. Hemolytic anemia, this is caused by destruction of red blood cells. And also, anemia may be due to kidney failure because, remember, the kidneys actually produce that hormone, erythropoietin, that is responsible for producing red blood cells. Leukemia, this is a blood cancer that's characterized by an abnormally large number of white blood cells. There are two different types. They can be acute or chronic, and generally people are treated with leukemia through radiation or chemotherapy to typically destroy abnormal stem cells, and then they will get a bone marrow transplant so they can actually produce more blood cells post-treatment. Multiple myeloma, this is a cancer involving the proliferation of plasma cells in the bone marrow, and this actually is a weakening of the bone by that particular cancer. Thrombocytopenia, this is an actual reduction in platelet number. This could be due to anemia, leukemia, a viral infection, someone that has been exposed to radiation, or a reaction to certain drugs. What this results in is unusual bruising and bleeding because now they're going to have a problem with the process of hemostasis.